Assalamu alaikum everyone. Welcome again to the microbiology classes. Today we are going to discuss another very important topic and the title is Scope of Microbiology. So let's start. As we know that microbiology deals with the study of microbes such as bacteria, virus, fungi, protozoa, rickettsia and algae. So it's clear that life certainty would definitely be questionable if there would be absence of harmless organisms. Harmless organism means useful organisms, useful microbes, useful bacteria, for example, because these useful or harmless microorganisms convert complex material into simple substances, as a result guarantee the life certainty. Definitely, the microbiologists, they are very crazy about to know about the mechanism that affect human, how microorganisms affect human existence, and this interest of the microbiologist has been the foundation of microbiology. Fermentation process, as we do know that in the earlier era, the mankind were only familiar with certain processes such as fermentation of wine, juices, milk, etc. Yet why these changes occurred were unknown until Pasteur, via his very famous experiment in the last part of the 19th century, showed a skeptical world that microbes are actually responsible for the fermentation process. And without microbes, fermentation is not possible. These microbes, for example, yeast and bacteria, they convert sugars to alcohol and acid, and this is actually the process of fermentation. So fermentation, it is carried out by ferments, better to say microbes. Without microbes, participation, fermentation is not possible. So in fact, microorganism has had a decisive role in shaping the world history. Emergence of pharmaceutical industry. The recent decades are known as an era of emergence of pharmaceutical industry. And as the name is self-explanatory, pharmaceutical industry is a place where pharmaceutical products are manufactured. But there are variety of the dosage forms of the pharmaceutical products. These pharmaceutical products, they are manufactured in the pharmaceutical industry, but then they are marketed for the public to be used. So the most important point is that there must be certain quality control tests that should be performed to get the pharmaceutical product of qualitative nature. Due to this reason, there is stringent regulation of manufacturing process so that the product which is manufactured in a tidy situation, it should be of qualitative and it should be marketed to the public, and it must not contain any microorganism. As we know, there are different kinds of pharmaceutical products. These pharmaceutical products are routes of administration, but when we use the term sterile products, they are used for those products which are given through the parenteral route. The parenteral route are also known as intravenous route because these sterile products, the first most important criteria as far as the quality of these sterile products is concerned, that there should be no living detective microorganism in the sterile products. As the name sterile indicates, absence of microorganism. It means that sterile products are those products which do not contain living microorganism of any kind. Therefore, we can say the sterile products should be able to pass test for the sterility. So for the sterile products, the quality control tests are also designed, and the most important quality control test is called sterility test. But who will perform this test? A skillful trained person is required for this test. So that person who has the basic knowledge of all these sterility procedures, as well as must be capable to interpret the results of the sterility test. No doubt, the pharmacist is the one person who has the basic knowledge regarding the sterile products, regarding the sterility test, and also is capable to show the interpretation of the results. So we can say in the pharmaceutical microbiology, the pharmaceutical pharmacist is important as far as the performance of sterile product test, which is known as sterility test, is concerned, and the interpretation of the results of the sterility test is concerned. It is obvious that the medicine contaminated with potentially pathogenic disease-causing microorganisms are a safety hazard. We can say that if medicines they are contaminated with the disease-causing microorganisms, so we can say these are called contaminated products. Then these are not called sterile products. Sterile products means absence of any kind of microorganism. So if the medicines they are contaminated with disease-causing pathogenic microorganisms, how can we assure that these are effective? Rather, efficacy, they will produce damage to the body. So because these sterile products, as I told you, that they are given through the parenteral route, they are given through the intravenous route, they are given in the form of injection. So these injections, these are called vulnerable routes or vulnerable areas of the body. For example, eyes, these are the mucosa. So if they are given in the form of injection, or if they are given in the mucosa, for example, eye, and if they are not sterile, if they are containing potentially pathogenic disease-causing microorganism, so instead of giving benefit, they would be disastrous as far as the body is concerned. So we can say that the sterile product should be manufactured under tidy environment so that there must be rigid exclusion of each and every kind of microorganism, and in order to detect whether the product is sterile or not, sterility test is performed, and this test is performed by a skillful trained person who is definitely a microbiological pharmacist, because he is the only person who has the basic knowledge about the sterility test, as well as he is capable to interpret the results of sterility test. Now the manufacturing of sterile products. We are now clear that sterile products means absence of microorganism in such products, and these products are administered only through the parenteral route in the form of injection through the IV route. But how will we manufacture these sterile products? The manufacturing of sterile products is totally different as far as the manufacturing of other pharmaceutical products are concerned. However, there are two different strategies which are used for the manufacturing of sterile products. The first, the most important one, that the product is made, then packed in its final container. Final container means that the container in which it is marketed, the container in which it is supplied to the end customer, the container in which it is supplied to the public. So this product is manufactured and then packed in the container in which it is to be marketed, and then this container which contains the product, it is subjected to sterilization either by heat or radiation or other means. And this selection of the method, heat, radiation or other, it depends upon the nature of the product. If the product which is packed in the final container is heat resistant, then we can sterilize it by heat. And if it is heat sensitive, if it is damaged by the heat, then we cannot use heat, then we have to use radiation or any other suitable means of sterilization. This is the first most important, generally accepted preferred method to formulate or to manufacture the sterile product, that the product is manufactured and then packed in the final container in which it is to be marketed, and then it is subjected to sterilization either by heat if it is heat resistant, or radiation or other means if it is heat sensitive. The second method, which is also called the alternative method, is that all the ingredients which are used to manufacture the sterile products, individually they are made sterile, and after the sterile ingredients are mixed under condition that do not permit the entry of microorganism, and this is the second method. However, the first method, it is generally preferred. Sometimes second method is also used. So there are two major methods of manufacturing sterile products. First method, the most important, the most preferred method is that the product is made, it is then packed in the final container in which it is supplied to the public. Then it is subjected to sterilization either by heat, if the substance is heat resistant, or radiation, or any other suitable means if the substance is heat sensitive. However, the second method, alternative method, is also there. All the ingredients used in the formulation of the product, individually they are made sterile, and then they are mixed under a tidy situation so that there is no entry of the microorganism from outside. So these are the two methods. Spoilage of the medicine as a result of microbial contamination, because we know that if these sterile products are not sterile and if they are contaminated, then it leads to spoilage of the medicine. It leads to destruction of the active ingredient, and it has one most important drawback, that it produces economical burden as far as the patient is concerned, because patient has to patient purchase the medicine. So it is economical burden, because if the medicines are destroyed, if the medicines are deteriorated, if the medicines they are spoiled, then instead of giving benefit, they would definitely be produce harm. And as a result, since patient has to pay, so then there is economical burden. So these are the manufacturing of sterile products, and this is important to know. Microbiology of 20th century. Microbiology is one of the newer sciences because science dominates the 20th century. As a result, microbiology becomes dynamic, exploding, and revolutionary. And we can observe that in the 20th century, the microbiologists, they were able to discover vaccines for the prevention of diseases such as smallpox and polio, and the drugs such as penicillin for the treatment of disease. So this is the golden era of the microbiology, that during 20th century, the dynamic, exploding, and revolutionary action of the microbiology appears, and as a result, the microbiologists were able to discover the vaccine as well as to discover the life-saving antibiotic known as penicillin. Similarly, the discovery of some of the vitamins and other basic food materials has resulted from the study of microorganisms. Certain vitamins we can obtain from the microbes, certain basic food due to the nanotechnology and biotechnology of the microorganisms are used in the biotechnology. So certain basic food material and vitamins are also on the part of the discovery as far as the 20th century microbiology is concerned. Similarly, we know that the body cells, they have their special chemistry, and also we know that there are certain parts of the cells which can pass on information from one generation to other generation. This is called heredity. So it means that heredity is also depending upon different portions of the cells as well as the chemistry of the cell. So all the knowledge regarding chemistry of the cell, all the knowledge regarding the influence of certain cell parts as far as the hereditary material is concerned. So it is also on the part of microbiology during 20th century. The biologist may use microorganism as possible source of food and oxygen for the space travels. So we can say that this microbiology during 20th century is not confined to the Earth's surface, but in the space we can also found its applications. The physician needs knowledge of microbiology to fight disease. So, because physicians, they are able to diagnose the disease, but how to treat the disease is actually the part over which the excellence of pharmacist is acknowledged. So since we are dealing with the manufacturing of pharmaceutical product, so the physician needs knowledge of the microbiology to fight disease. For example, this is a vaccine. as far as flu is concerned. So don't spread flu. This is the message. We have this vaccine. As a result, the flu, it could be minimized, it could be controlled. Similarly, industrial scientists often utilize microbiology in the manufacturing of useful chemical products. The geologist frequently uses information about microorganisms in his search for oil. The geologists, they also utilize the concepts of microbiology as far as searching of oils is concerned. Thus, microbiology is important from the core of the earth to the far reaches of outer space. So it is not confined only to the upper surface of the earth. It penetrates deep into the core of the earth, and also it has the application as far as space is concerned. So this actually become possible during 20th century. Now move towards the diagnostic application. This part is very much important and needs proper focus. First one is isolation and identification of microorganisms from the pathological lesions, prognosis of disease, drug of choice selection, and identification of source of infection. So let's start with first one, isolation and identification of microorganisms. You know that different microbes, they are responsible for different types of diseases, and when we know about the nature of the microbe, then we could be in the much better position to treat them, because these antibiotics, they are not equally effective for all the microorganisms. There are certain antibiotics which are effective for the gram positive bacteria, and they are ineffective for the gram negative bacteria. Similarly, there are certain antibiotics which can cover only the gram negative bacteria, and they have no effect on the gram positive bacteria. So in order to know about proper treatment protocols, we first must know what is actually the nature of the microbes. So in that case, in order to diagnose them, first of all, the most important thing is that isolation and identification of microbe. When we identify the microbes, then we could at least treat them rationally. So this skillful task is accomplished by screening blood sample as well as urine analysis to diagnose any infection which is prerequisite for rational treatment. As I told you, that treatment could not be successful if there is not proper diagnosis. So for the diagnosis, we must identify the microbe, and for the identification, we need proper knowledge. So in that case, we have two important procedures, that either we can take the samples of blood from the patient, or we can take the samples in the form of urine from the patient. When we take blood samples, then we can culture on the solid media, because we have discussed in the previous lecture that on the solid media we get colonies, and these colonies actually represent the pure form of the bacterial growth. Now when these bacteria, they are grown on the culture media, and also solid culture media is of different types. For example, McConkey's agar media, for example, nutrient agar media, for example, blood agar media, for example, chocolate agar media. There are different types of the media, and these bacteria, they need different media. Some bacteria, they grow on the McConkey agar media. Some bacteria, they grow on the blood agar media. Some bacteria, they grow on the chocolate agar media. Some bacteria, they grow on the nutrient agar media. So these are the different petri dishes which contain bacterial growth, and also you can observe the pattern of the growth. So this actually, when we take the blood and apply on the petri dishes of different types, we get different kinds of the bacteria, and then these colonies once appear, we take these colonies, and then we perform certain test, or we perform staining procedure, and then identify under microscope. In this way, when we identify the microbes, then this is very much important. As a result, rational treatment is possible. This is the first case how the blood samples are treated. Blood is taken, subjected to the petri dishes which contain different types of the culture media. Each culture media facilitate the growth of particular type of the bacteria, and once these growth in the form of colonies appear, with the help of certain tests, we can identify the types of the microorganism. On the basis of this we can confirm that this disease is due to this particular microorganism, and then we can suggest a particular antibiotic for that particular microorganism. This is isolation and identification of microorganism with respect to blood sample. Now move toward the urine sample. Mostly urine is taken to identify the urinary tract infection. Different microorganisms, they are responsible for the urinary tract infection. So for this reason, first of all, the urine sample is taken from the patient, then it is centrifuged. It is subjected to centrifuge machine. As a result, it is centrifuged to capture the bacteria. This centrifugation process, it capture the bacteria, then it is subjected to the Raman spectroscopic cups in which the bacteria, captured bacteria is present, and then as a result, information is generated as a fingerprint, and in this way we can find out. For example, this is Escherichia coli. It is the most important cause of urinary tract infection, particularly in case of females. Similarly, Escherichia fecalis, this graph is responsible for that one. So we can then, by observing the graphs, we can also know about the name of the bacteria, because I told you that these bacteria, they respond differently to the antibiotics. So for the E. coli, different antibiotic is there. For example, Escherichia fecalis, different antibiotics. So this is the urine analysis, and this urine is taken, it is then subjected to centrifugation, then to capture the bacteria, then we perform this Raman spectroscopic analysis. This finger type graphs are obtained, and by measuring the graph we can identify. So this is the isolation and identification of the microorganism from the pathological lesion, either by taking the blood sample or by taking the urine sample. Now moves towards the prognosis of the disease. So what is meant by prognosis of the disease? Prognosis means to forecast the course of disease, to know about the future of the disease, to know about whether disease is progressing or disease is controlled by the application of the antibiotics. So the concepts of microbiology provide sufficient information about prognosis of disease. Sometimes we perform tests, and by observing the test, we can find whether the disease is controlled, whether the disease is reduced, or disease is progressing. For example, the best example in order to understand the prognosis of disease is the Vidal test. Vidal test, this is the name of the test used to find out, to diagnose the typhoid fever. Now, in this case, in Vidal test, rising antibody titer signifies active disease and ineffective treatment, whereas falling titer indicates effective treatment and curing of disease. So let me explain with the help of this one, because these are specific terminologies used in the microbiology. So you must know Vidal test is the test used to find out the typhoid fever, and in this case we deal with the antibody titer. And if the antibody level, if the antibody titer, if the antibody amount, if it is greater, it means that the disease is active and treatment is ineffective. But if the antibody level is less, we can say that disease is not active and the treatment is effective. Now in this case, Vidal test, we perform dilutions. 4 to 5 dilutions are manufactured. For example, 1:20, 1:40, 1:80, 1:160, 1:320. This is the 5th dilution. This is the 4th dilution. Normally, we perform up to 4 dilutions. Sometimes 5th dilution is also in. Now, how these are generated? Now, what is 1 and what is 20? You know here, this is Vidal test. In the dilution, this is tube 1, this is tube 2, test tube 3, test tube 4, test tube 5. So the salmonella antigen, because the typhoid fever is caused by salmonella typhi, so yeah, salmonella antigen, either O or H. There are, because there are different types of antigens as far as bacteria are concerned. In case of Salmonella typhi, one is called O antigen. O antigen is called cell wall antigen because you know that the outermost layer of the bacterial cell is called cell wall. So this cell wall is also acting as antigen, and it is called O antigen. When Salmonella O antigen is added, so it means that actually this is the cell wall of the Salmonella typhi. However, there are certain bacteria which are motile due to the presence of flagella. Flagella is also antigen, and H antigen means flagellar antigen. So if O antigen are added, it means cell wall is added. And if H antigens are added, it means flagella is added. Okay, you clear it. Now this is taken, and because from the patient we need blood, so the serum of the patient is taken. Now this is 1 part and 20 part. 1 part of the antigen and 20 part of the patient serum. For example, 1 microliter of antigen and 20 microliter of patient serum. Then in the second dilution, we make 1:20. Again, antigen is same 1. In all the dilutions, level of antigen is constant 1, 1, 1, 1, 1. But the level of the patient serum in which antibody is present, it is different. In the first, it is 20 microliter, and in the second, it is multiplied by 2 of the first, 20 multiplied by 2, 40. Then in the third, 40 multiplied by 2, 80. And in the fourth, 80 multiplied by 2, 160. And in the fifth, 160 multiplied by 2, 320. So this 20, 40, 80, 160 or 320, this is actually the patient serum which contain antibody here. Then we check whether when we mix 1 part of antigen with 20 parts of the serum, whether agglutination occur. If there is agglutination, we proceed here. Still the test is negative. There is no assurance that the patient is having the typhoid fever. We move towards here. Again, 1 part of the antigen mixed with 40 parts of the serum, and we check for the agglutination. If there is agglutination, still we move towards the third part because there is again no assurance that the typhoid is present or not. In the third dilution, if 1 part of the antigen is mixed with the 80 part of the patient serum, and we check for the agglutination. If agglutination is present, still test is negative, we move towards the fourth dilution, and we mix 1 part along with the 160 part of the patient serum and check for the if the agglutination in the fourth dilution is there, then we say that now the patient is having the typhoid, it is positive. And we normally stop here the test because fourth dilution is sufficient. If in the fourth dilution still agglutination is present, we call it positive, and as a result we signifies that the patient is having typhoid fever. However, sometimes fifth dilution is also made, and when we mix 1 part of the antigen along with the 20 parts, 320 parts of the serum, we check for the agglutination. If the agglutination is there, it is super positive. Okay. Now, so agglutination is noted in these dilutions by mixing antigen along with the, because antigen and antibody react with each other, and this reaction is actually called agglutination reaction because here the blood is used. When blood is used, then this is called agglutination, hemagglutination. So if here agglutination occur, it is negative. If here agglutination occur, negative. If here agglutination occur, negative. But if here agglutination occur, since it is required that 4-fold dilution is mandatory, so it is positive. Sometimes we do perform in order to check super positivity. So fifth dilution is positive, then super positive, it is chronic infection. Now, since up to 4 dilution the test is positive, so this is called titer. Titer is called level of antibody in the body. Level or amount of the antibody in the body is called. Now if let's check the prognosis of the disease. If you get the result typhoid fever, Vidal test, and if you check that up to 160, it is positive, so it means active disease is present and ineffective treatment is there. But once you start antibiotic, and then again after 10 days or after 7 days or after 14 days, again you perform the same Vidal test, and now if up to 140 or 180 agglutination occur, but if there is no agglutination at fourth dilution, so we can say up to third it is negative, so we can say that it is now the titer is falling, and it indicates that there is effective treatment and curing of disease. So this is prognosis of disease. We can observe the course or the future of the disease, whether it is progressing or it is reducing. So if the titer is increased, then we can say active disease is present and treatment is ineffective. However, if the titer is reduced, if the fourth dilution is not showing agglutination, and up to third dilution or up to second dilution agglutination is there, we say that the patient is having no typhoid, now there is no active disease, and the treatment is effective. So this is prognosis of disease. Drug of choice selection. So it is very important to perform culture sensitivity test. But in Pakistan, unfortunately, we see whenever we become ill, we either go to the physician, or sometimes we don't go, we directly go to the medicine shop and ask for the antibiotic and start using the antibiotic. This is not the right way to use the antibiotics. Even when we visit the physician, it is the responsibility of physician first to perform the culture sensitivity test, because as I told you that when we suffer from any infection, it means that microorganism is present. So first we must know what is the nature of the microorganism, because I told you that all the antibiotics, they are not effective for all kinds of microorganisms. There are certain antibiotics which can cover the gram-positive infection, and they have no effect on the gram-negative bacteria. Similarly, there are certain antibiotics which only affect the gram-negative bacteria, but they don't have the gram-positive effect. Similarly, there are certain broad-spectrum antibiotics which can affect both gram positive as well as gram positive, gram negative bacteria, but widespread and unneeded use of these broad spectrum antibiotic, it can lead to the drug resistance. As a result, the same broad spectrum antibiotic even cannot work. So the best way is that to note about the drug of choice, we have to perform the culture sensitivity test. In this case, the blood of the patient is taken, and on the solid culture media, it is grown, and as a result, colonies appear. This is called bacterial growth. Now, different antibiotics. The discs are available. These are called antibiotic discs. Here, for example, A, B, C, D. These are actually the uh, uh, terms used for the antibiotics. A antibiotic, B antibiotic, C antibiotic, D antibiotics. Then on the growth, on the colonies, we place these antibiotic discs. One antibiotic which is labeled as antibiotic A, it is placed here over the colony. B antibiotic disc is present here, C is here, and D is here, and then it is incubated, and at 37 degrees centigrade or whatsoever the temperature is, and after specific time, we note how much area is cleared by the antibiotic. You can observe the color here. This color, it means that bacterial growth. Here it is white. It means that the bacteria, they are removed by the antibiotic A. Here, this much area is cleared by the antibiotic. It means that antibiotic B is clearing this much area. Antibiotic C, no white area. It means that there is no effect of this antibiotic as far as the bacterial growth of this petri dish is concerned. And antibiotic D, the mega area is cleared by this one. So we will know this area, this clearance area, this is called zone of inhibition. This area which is cleared by the antibiotic A is called zone of inhibition of A. This is called zone of inhibition of B. This is called zone of inhibition of D. But here in case of C, there is no zone of inhibition. So then when we apply different antibiotics on the growth, we note the zone of inhibition. Then we lastly note which antibiotic produce larger zone of inhibition. A, B, and D. Out of these three, D is producing larger zone of inhibition. So that antibiotic which produce larger zone of inhibition, that is called the drug of choice for that growth. So here, zone of inhibition produced by A, produced by B, produced by D, but not produced by C. It means it is ineffective antibiotic. bacteria are resistant. Here bacteria are not resistant. It is effective. It is also effective. D is also effective. So we can say that the bacteria are susceptible to antibiotic A, susceptible to antibiotic B, and susceptible to antibiotic D. But if we use A, B, or D, we can eradicate the bacteria. But if we use D, since it has produced larger zone of inhibition, so we can say that this is the drug of choice. If we use this antibiotic D, we can quickly, rapidly eradicate the bacteria, and the patient is quickly recovered from the disease. So by using the microbiological knowledge, drug of choice could be selected, and this can be done by culturing microorganisms and performing drug sensitivity test. Thus, we can gain guidance in the treatment. Identification of source of infection. Sometime in sudden outbreaks means epidemics. उस दिन भी मैंने आपको बताया था कि एपिडेमिक है कि किसी एक एरिया में एक ही टाइम में बहुत सारे लोग बीमार हो जाएं. फॉर एग्जांपल आप हॉस्टल में हैं, आपको डायरिया हो गया, बहुत सारे लोगों को डायरिया हो गया. दिस इज आउटब्रेक और एपिडेमिक. So in sudden outbreaks, we have to find the source of an infection. So this source of infection could also be found out by using the microbiological concept. For example, salmonella infection. किसी एक एरिया में, एक कम्युनिटी के अंदर एक ही टाइम में अगर बहुत सारे लोगों को टाइफाइड हो जाए, तो हम उसको देख सकते हैं कि वो कैसे हो सकता है. So there are three possible sources of infection. For example, the cattle, because we use the milk of the cattle. If the cattle is having the salmonella infection, and if we use unpasteurized milk, raw milk, बाज़ औक़ात वो जो है ना देहातों में दूध को गर्म भी नहीं करते, इसलिए कहते हैं दूध गर्म करें ताकि उसके अंदर माइक्रो ऑर्गेनिज्म मर जाए. जैसे वो उसको उससे जानवर से लेते हैं, फौरन उसी टाइम पे उसको पी लेते हैं. तो ऐसी सूरत में कच्चा दूध जब पीते हैं, सो माइक्रो ऑर्गेनिज्म इज प्रेजेंट, एंड एज ए रिजल्ट बहुत सारे लोग वहां से इन्फेक्शन हो सकते हैं. द पोर्क, pig meat, pork, if you eat, and if let's suppose this pig is having the salmonella, we are Muslims, so we don't eat, but the other peoples who do eat, तो उसके लिए भी ये है कि अगर ये इन्फेक्टेड है और इफ इट इज नॉट प्रॉपरली कुक्ड, तो ये भी सोर्स ऑफ इन्फेक्शन है. समटाइम्स बाय टेकिंग एग्स, अगर आपके पेट्स जो हैं वो contaminated हैं और अगर आप eggs यूज कर रहे हैं, so in that case, if in any community, if the salmonella outbreak occurs,